at the onset, I must first start by thanking India Science Fest for hosting this very nice program that I have an opportunity to go through your program and see how young participants are contributing in various science-related topics. It is really a pleasure for me to introduce a topic that is not so well known in India. We are growing, but we are still quite behind than the rest of the world. So I thought that this could be a very important topic that we should introduce to our Indian community, Indian science community per se. And there should be, because we like to see more enthusiastic participation in this field of research, which is which can come from multiple subject areas. So as I go through my talk, you will realize what I'm actually trying to say. So it's not just, even though I am an application biologist, but now my research involvement is in the medical imaging, more in the experimental sector, since I'm not a doctor, so I don't do the medical imaging for humans, but my experimental patients, which are mouse models. So in tiny animals, we do the same imaging things that doctors do in the human patients. So let me put out my slide first. So thank you, Science Fest, for giving me this opportunity and thinking about this topic as part of your program this year. I'm also really thankful, and I wish that all the participants who are present here after that terrified year of 2020, you will have a lot better, joyful, and successful year ahead of you. And the first thing that I want to say about visual. So, what is a visual? Why visualization is so much of an importance? So, visual perception is a vital cognitive processing. Okay, so you mean that of all the different sensations, the having an eye is really a fortunate thing for us who have these, and for some reason who have lost that. So so it's a thing that actually make you get a very deep sense of learning. So you may think of a common example and take from your everyday life that you are reading a book. So what is going on when we read a book? We are just looking at words of a book and being able to make sense of the plot of the story, and which is basically I'm calling as visual perception at work, right? So also think about an attempt such as, end of December, there was a celestial event that Saturn and Uranus has come closest proximity to the Earth. And we were all trying to, reach out and try to see. But, people who have a high profile binocular or a telescope, they could witness it a lot better than some of us who may have just tried it with our little mobile phone. So the stars which came through close to the Earth, which is like a glorified event for the Earth, we may not see it, we may not have another chance to witness it in our lifetime. And we find that we are limited in terms of actually experiencing how they look and what they look and all that thing, right? So this is where the visual perception comes, and it actually make you, either you jump up, and yippee and the full of joy when you actually can experience it, or you can get sad and come on, I am no good, I don't have any tool to witness it. So something like that. So these kind of repercussions, so that immediately tells you that how this cognitive sense is actually making an effect in your learning process. So when it comes to the human body system, so if you imagine the body is basically an opaque system. So now all the normal function or even the disease function, so how it is the body has been adapting inside this opaque system, which is from outside we cannot actually realize. But there is the body system is have the bony structures, we have nerve structures, we have muscles, we have different organs, we have blood vasculatures, and so many other complex organ systems that makes our body run and makes us run. So now anything that goes wrong repercute into the disease, and diseases are of many types, although in my talk I will be mostly focusing on cancer. So, so what is a cancer? So, although I suppose that most of the audience would know this, not necessarily that the deeper insights, but in our neighborhoods, in our families, or even our close associates, we often come through this terminology, cancer, and some person has got cancer. So what is really cancer is? So cancer actually it's still the leading cause of death. The coming out of the corona year, I can tell you the corona is definitely a very infectious disease, and it has killed quite a large number of people around the world, but still nothing in comparison to the death that caused by cancer or heart disease. So they are actually the leading cause of death in the world. So what in case of cancer, what happens is, so if the you and me, we are all different. How we are different? We got inherited a set of genes, and as that combination of genes that I have got in me is different than the combination that you have got in you. And now when these genes actually work in you, how it works and how their aggregated effect of the function, that makes you different, how you function than I function. So basically the cell and all these genes are inside the cell that we all know, right? So now these DNA gene structures, there certain change happen due to various factors. It could be a virus, it could be an environmental pollution, it could be many factors, or even it could be just an inherited mutation. So when a mutation happens in a gene, so that cells start functioning differently, and that's the beginning of the cancer. And at that stage, it was not detectable because it was just happening in one cell. So essentially, all cancer starts in one cell. And over the time, so there is a huge time gap. Sometimes that time could be 3 years or 3 months, or even 30 years, 40 years in certain cases, in certain types of cancers. So this time is unlimited time, and then there is an outcome that happens, which is actually a change in the structure. So it's a very neat balance where the normal cell system works, and then because of the change in the function, ultimately it enters into a state where there will be a change in the structure. Change in the structure meaning change in the anatomy of the body. So then when you compare with a normal anatomy to the diseased, you have to catch up where things have gone wrong, where there is a change that has happened. So these are all that imaging can actually help. And if you look at the history, you will see that imaging science has a long history, at least about over 100 years by now, and 120 years by now. So even though it was introduced with, the beginning was the electromedicine, which is more for therapeutic use of electrical or electronics as a medicine, but later on it evolved into the field like radiological diagnosis. So in radiology, there are a lot of different toolbox that actually came over the period of time. And one of the things that ultimately on last 20 years or so, what we have achieved is an incredible power to visualize our opaque body system. And we can actually make the body, treat the body like a transparent system. So that we can see inside all the changes that is happening. And that is more of a functional catching of how the molecules are differently functions inside the body. And that is what this medical imaging is now called as molecular imaging as well. So the intriguing means, in science, there are all kinds of stories, some crazy thoughts. That's what makes, the inventions. So this is a person who I should assign that he is the game changer in the field of medical imaging. So this Charles D Heavies. So he was a Nobel laureate in 1943, and he has great histories, like great stories, like at that period of time, the Nazis actually wanted to snatch his pure gold Nobel medal, and because he was a chemist, and he actually discovered this aqua regia, which can melt the gold. So he just put to save his gold medal from the Nazis. So he actually put the gold medal in this liquid, and then this liquid gold, he kept it on itself. So that is of no use for anyone. So that's how he saved his gold. Later on, post-Nazi era, he actually gave it, the gold was extracted back from that liquid, and a new medal was made. So that's how, that's the story that is well known in the West. You can read about it. It's very intriguing. But the story that I want to tell you, and this should be the beginning of the talk, about how we can track molecules. So that's where actually he was a radiochemist. He was a radiation chemistry person, and he had access of radioactive salts. And this, as the cartoon suggests, that he had a suspicion that the landlady's house where he was living, and whatever his leftover food, it is being utilized by the landlady to make the food for his next meal. So what he thought is he had these radioactive particles in his possession, and he also had scanner to monitor where to find. So what he did is his leftover food he mixed up with his radioiodine salt, and then he left. And next day, first he scanned his plate, and he found that yes, indeed, it is radioactive. So what essentially this craziness has evolved is shown us is the ability of radioactive molecule to track material over a period of time. So that become the principle of all the medical imaging that we are doing today. So this is really a game changer. With that note, I will now like to just explain you little basics about what is image and how we differentiate a good versus a bad image. So the visual cognitive action. So it like the image I have placed on the left side. So there is a person, but if the image quality is of this type, so naturally it makes hardly any sense. You cannot figure it out who it is, what is happening to him, or why his picture is in front of me. So you can't really make out. But when that person is actually, so this image is a lot more meaningful to you, because here you can vividly see. So how these vividly see means what? So there are basically these four bullet points that I'm using here: is the resolution, sensitivity, specificity, and the field of view. So resolution is the main thing. Like the sensitivity, as the sensitivity increase, the resolution also naturally increase. So with more and more sophisticated instruments like our TV or the digital camera, or even the digital camera on our mobile device, so they all are evolving, and they're being able to resolve lot more finer details within our pictures, right? So that is what is the spatial difference. So for example, even on this image, I cannot resolve where the eyelids are. And so the camera that has been used is not good enough to differentiate the the eye, individual lashes on the eye. So that's the spatial resolution. But there are camera now that you often see images in your TV, that where each eyelash can be nicely means you can differentiate, right? Then there is also the resolution of another type, is a temporal. So over the time, how well I can, so just like the video versus a still camera. So the video is capturing my image on the real time as I am talking. It's not that it is getting stuck at some point and you are missing out. My lips and my words are not matching. Sometimes it happens in movies, right? So, and it looks odd, right? So that is sort of an example of a temporal resolution. And also there are spectral resolution. So now the color, so there are diseases like jaundice, where the skin gets a yellow tinge versus your normal tinge. So those can my imaging device separate that spectral difference? So these are all the parameters that can define which instrument to use for what disease, and that is how we go. And there are, of course, the specificities, like if I'm trying to can I catch the true positive over true negative? So that's the specificity. So more the device is ditchful, it's a bad device. So that needs further improvement. And then the field of view is like whether I want to see the whole body versus half body, or just head, or even think about you walked in your eye doctor's chamber, and he will then focus with his microscope only at the cornea inside your eyeball. So the device, what you want to see, and then what it can resolve for you, so that you can actually have a vivid feeling of the normalcy or the disease state, right? Am I getting clear? So moving on, so if you have to, so one of the aspect, although this field is very, very multidisciplinary in nature, of physics, you will get a sense that the people from physics background, engineering background, chemistry background, they all, or even computation background, they all can come and contribute in making different tools, making different probes that is required to use those toolboxes, which are the medical imaging device. Or even the biologists and clinicians, they are no less here, because after all, is the biology, which is the battleground, and which is the application field, where all the challenges are how our understanding has evolved to what to challenge, and then what device to do or what probe to make, so that can get defined. So coming to the probing, so if you have to see something, you need, majority cases, you need a probing. And this is where actually we get this very basic science of our 10th, 12th board. We all know that the shorter the wavelength, higher the energy, higher the wavelength, shorter the energy. So this inversely proportional relationship between the energy and the wavelength, and that's where you can see the different medical imaging devices that have been defined, where PET and SPECT are on the high energy, utilizes high energy emissions, and then our visualization area is only this much, which is very narrow belt of 400 to like somewhere around 350 to 750 nanometer range. But beyond then, on the lower, longer wavelength area, we have Raman spectra, then the RF, the radio waves, and the FM and AM and so on. So, so in the visible area, which is I'm starting with that because that's the area we can realize most, because this is what we can visualize. Our eye cannot perceive, our eye perception is only limited to this area of colors. So, so there we have multiple different probes that can form, and I suppose most of you know what is a fluorescence. So when any moiety that can be thrown with some light, and then it can get excited, and then when it relaxes during that state, it release the farther red wavelength light. So that's the fluorescence. And then there are, and some of these fluorescents which are on the blue or the violet range, they do not really work that well in a body system because there are a lot of absorptions. And we have a very vulnerable condition because of the presence of hemoglobin, which actually absorbs a lot of light. So the imaging using the fluorescence in blue or green range is a challenging job, but it works very well when any wavelength above 600 to 800 nanometer range, where lots of different other dyes are also available. And there are also fluorescent proteins of multiple colors that can be available, and this helps us to do multiplexed imaging. So by separating the different color, we can find the different source, we can level different source that we simultaneously can monitor using such kind of light emitting probes. And there are other classes, of course, like luminescence, where it is not at all that you need to excite with some light, but it's an enzyme substrate reaction in most cases, and that produces the light. So it's the best option, but it's the very weak light. Photons amount that comes out are very weak. And so sometimes catching that, we have to have a device that actually can work on this. So all these light emitting things works very well in the area of optical imaging, and more used in the experimental science. There are also these nuclear decay, which is actually Charles D Heavies. So there are a couple of different types, like the gamma decay or the positron emission decay. So here is what actually the positron is an antimatter. So it's like a positively charged electron. And when it collides with an electron in the neighboring molecule, so it actually makes two opposite direction radiations, which are called positron. And that can be and because of their this clear principle difference. So it can be easily computed, and that's what has given rise to the toolbox that is called positron emission tomography on today's date. The gamma rays are, for a long time, it is in use, and it is called single photon emission tomography or SPECT system. So what is the difference, basic difference, if you want to see about the radioactive versus optical tools? The radioactives are always on. They are very much quantitative because they are high energy. There is no absorption or deflections ratios. These coefficients are insignificant, and they are human related. So it can be used in bigger body size. Versus optical systems are more for experimental sector, as I already mentioned. It can be turned off and on. It can be of high sensitivity. And because of the spectral separation, we can also do multiplexing. Here, they all emit the same energy. The one molecule emits. So until and unless you have the ability to energy resolve, uh, you cannot actually multiplex the PET probe or the SPECT probe. Another good thing I want to, main point I want to mention here is that this time wait. So because it is always on, so whether it has a bound probe or an unbound probe that is circulating in the body blood, it cannot be differentiated. So we need to give the wait time so that all these excess probes which get eliminated through kidney or get eliminated through the urine, and then we can find a better contrast where the bound probes are on the disease location. So this is a very important aspect. It is called incubation time. So another aspect that I want to talk about, how now the different signatures of material that can be used, as I am saying, whether it is an light emitting probe or whether it is an radioactive probe. But there has to be a target where in the cancer we do this a lot, that we find out a carrier, and then with the help of chemistry experts, we can actually tag a level based on the suitable imaging system that we have. And then it turns as a probe or a warhead, and that can actually target to the molecule which is on the disease location. So this is how the probe can be generated and utilized. So here is the experimental mouse where the tumor is placed, and then we developed a new probe using such a small peptide tagged with a fluorine 18, which is a PET positron emitter, and is become the PET probe. And when we replace this tag with a Alexa fluor, for say, then it can become suitable for optical imaging, and it is like an more utilized for experimental sector. There is also another way now, as the field has emerged, it has opened up a lot of options, new options. So some of these probes, so like this phenomenon, like Serenkov luminescence imaging. Again, this is another scientist who discovered this phenomenon. It's a physical phenomenon. It is generally witnessed in all radioactive, producing areas. So there is a luminescence light halo that glows around the radiators. And this is basically the charged particle like the electrons, when they travel in a very fast speed, higher than the speed of light, in a dielectric media like any watery media, like our body is also 70% water. So there is, it can also be considered as a dielectric media. So this electron, when it collides in a dielectric media with, then it produces these photons. And how you detect that? The photons can be proven by covering with a black paper, but radioactive you just cannot cover with one black paper. And so this CLI image, which is really speaking luminescence or the optical in nature, and the same probe for the same mouse can also be used on the imaging system, which is a PET scanner. So there are versatile probes that are already in place for different functions. As I was telling you, that cancer, there are a lot of different functional changes, something like tumor metabolism, tumor proliferation. So cancer cells are different in terms of either the cellular metabolism or the proliferation or their death mechanism. Because of all these things, there are now specific probes that has been made and that can utilize. And basically here we are, the image that we are generating are actually, we are probing, we are studying how the function has changed. I hope I make sense. And for the experimental sector, all the bigger, now the human body is a lot bigger size. Now resolving it from the mouse model is another set of challenge, because it is also an physiologic entity, but here all the organs are at least, uh, there is a very big difference in terms of their dimension. So now the same brain, the brain of a human being and the brain of a mouse, so there is a big size difference. So now the size is a huge constraint in terms of resolving power, right? So, and how far we can do that. So that is what was the challenge, but still we have now the whole array of imaging systems that are being used in the medical hospitals. We also have experimental sector, all the imaging toolboxes available, and their advantage, disadvantage is given. So coming to the medical imaging, the structural versus function, what I was telling you. So one quick example could be like magnetic resonance imaging. So MRI scan, doctor has asked you to do MRI scan, or for someone So what they are like to do? So they are basically, suppose there is a spinal cord injury. So they want to resolve the change that has happened, and they will basically, they have the register, the atlas registered in their mind, and when the radiologist look at the film, they will figure it out where there is a change that has happened, and that is how the disease can be detected. Versus in PET, it is like almost transparent, the body, and you can see where the new growth is happening, that is utilizing higher glucose or different probes that I've just shown you in the previous slide. And another very interesting study, I could not resist myself to share with you on this, that sometimes we can even combine different probes. So something like 18F FDG, which is a glucose. So higher metabolic rate of cancer cells can be imaged using FDG. So that's the gold standard probe. So this is a patient with multiple metastatic disease all over the body, and that's where all the different tumor, big and small, they can be visualized. But then they made it a cocktail of two different probes, and the other probe is actually sodium fluoride and the 18A version of sodium fluoride. So this is basically used for bone scans. So it could be an alternative to X-ray. X-ray also brings out the bony structures. So on the PET scanner, using 18F, you can also get the bony structures, unlike the FDG scan. And when you combine these two, you can actually get some of these new cancers on the bone surface, like the ribs. So these are not so much apparent, but they have now become a lot more apparent through the F18 sodium F18 scan. So this is called cocktail scan. So these are all all the different trials and errors, and the field has progressed quite a bit, and we have gained superpower in terms of visualizing and realizing how the cancer disease is evolving inside the body at different stage. And we are definitely not limited. So as I was mentioning you right in the beginning, that this field is very, very multidisciplinary in nature, just because that no one system is good enough to give you all the informations that you need. So that's where actually the field has evolved, and people have realized that multimodality imaging is a very necessary thing, and where actually the physics, mathematics, engineering people who come and who solve the problems in terms of building new systems. The computational science people who can also do, the through computer platform, you have separate machine, but you can have one common file tag like DICOM file, and then you can merge them together on a software platform through programming, and you can actually use that anatomic and functional references together. Multimodal agents, so something like chemistry can contribute, where multiple things with different signatures can be stitched together, so that one probe we deliver inside the body, and we can be scanned on multiple different systems suitable for each signature. So one example of the physics engineering contribution, more emphasized, like it is like a PET scan, CT scan, they all evolved separately. But over time, we realized that the PET-CT combination actually makes the image more specificity and sensitivity gain. So meaning a more accurate prediction of the disease can happen when we combine these. So something like PET-CT is now become a medically standard. There is no separate PET or CT system. Almost all hospitals get the PET-CT system, so which is a merged field. And now even there are MR compatible PET, so MR PET or even PET-CT SPECT. So these are the different mergers of different engineering marvels that is happening. And another example of classic example, I am citing this paper from Nature Medicine published in 2012, where the chemistry probe has been developed. So there is a multiple things, so different tags like DOTA gadolinium, which is a MRI probe. MRI actually whole body scan and identify the disease where it is. And then there is gold, which is the core inside, that is actually suitable for photoacoustic. So this is a system I haven't talked about. This is like you throw a sound impulse, and you actually excite with the laser, and you listen to the sound. So again, because the signatures are different, there is the background to signal is contrast is very high, and that makes the system very sensitive. So, and gold, it has this natural probe for photoacoustic. So these are nano-sized probe, and with a combination of gadolinium and gold, and there are also Raman chemical layers in between, and that makes it compatible to perform scans in MRI system or photoacoustic system or the Raman system, Raman spectroscopic system. So this is the shown that surgery guidance, how it can help. So the grossing was done by MRI, then photoacoustic is actually doing the surgery piece by piece. You can see in an experimental sector inside the brain, the tumor is growing, and they are taking out piece by piece, quarter, half, only 1/4 left, and something like that. And then when the remnant, the Raman is a beautiful system to do this, but Raman's limitation is it cannot penetrate too deep. So until it is resected, it cannot be this thing. So with that note, this would be my last slide, and this cartoon as there is a time in my life I have gone through this. So um, I started learning what is imaging. I never heard of what is imaging, CT scan, PET scan. Nothing of that sort was known to me at some point beyond the year 2000. And during when I moved to Stanford University in UCLA and UCLA for my higher studies, that's where I got exposed to this. And then I actually got mesmerized with this field in the growth, the impetus that comes from multiple different subject areas. So that was very, very intriguing, and it actually stayed me. So, and of course, we on today's day, this cartoon may be too old now. And post-COVID era, so this cartoon is absolutely invalid, right? So now no man even can say that you cannot do this for me. We have to do online. So I end with this nature heading, that is the medical imaging, removing the blindfold. It was published, it was a whole issue of Nature that has focused on how different medical imaging technologies are removing our blindfold, and technologies have taken us too far. We can actually we all know nowadays that immune cells. In the COVID time, we have come to know many things, and that is also one thing: clinical trials, immune cells. So these things we can actually track each uh, uh, each of the immune cells. So with that note, I thank you.
Thank you so much for all your time. Really thankful that
And
you took out the time, and we will hope to collaborate with you in the future again. Thank you so much for all your time.